Well, first of all, let me thank the organizers for the opportunity to speak at this event. And I must say that I've been very impressed by the extremely rapid progress in computational biology, both in terms of building up a base of very talented students, and also the highly productive research that's gone on here. I've greatly enjoyed the talks yesterday. And I'm lucky enough to see that the screen has come back. So I'm going to talk about a couple of problems. The first involving protein interaction networks, and the second involving reconstruction of signaling pathways. And let me begin with a few well-known facts about proteins. So the DNA sequences in our genomes code for proteins, which are essential to almost every process that takes place in living cells. Many proteins are enzymes that catalyze biochemical reactions and are vital to metabolism, and proteins have many functions in the cells, structural or mechanical functions, and also functions having to do with the conveyance of information from one cell to another and the response to signals that impinge on cells. So when we try to understand the regulatory mechanisms that take place in cells, we are to a large extent trying to understand how proteins interact coherently to make things happen. So proteins interact to form aggregates or collections of proteins that work together to perform functions in the cell. And so to try to understand regulatory mechanisms, we need to understand which sets of proteins work together. And in particular, for this purpose, we can look at a sort of graph theoretic structure that represents the proteins in a given organism and the interactions among them. And this structure is called the protein-protein interaction network of the organism. It's a network in which the vertices are the proteins of the particular organism, whether it's Homo sapiens or Drosophila or yeast or some other. And the edges in the network, in this undirected network, indicate pairs of proteins where there is some information to indicate that the proteins physically interact. And so in this network, if a set of proteins work together, then we would expect that that set of proteins or vertices would induce a connected graph. And so we would like to look at various connected subgraphs within the protein-protein interaction network of a species and gather evidence as to whether those corresponding sets of proteins have a common function. So one of the clues that we can use to identify sets of proteins that have a common function, well, one way to do it would be to to label the proteins according to their functional annotation in the Go hierarchy, for example, and then look at sets of proteins that have a common functional annotation. That turns out to be, because of the great variety of functional annotations, that turns out to be cumbersome, and so we look for other graph theoretic tools that might indicate protein modules, sets of mutually interacting proteins. And one idea is that if proteins are working together, then it's likely that they would have a high density of interactions among themselves, and that the set of proteins would be relatively isolated from other proteins in the cell, so that the number of interactions between the proteins in the set with the outside should be relatively small. This is a property that is sometimes referred to as modularity, And it's captured by a statistic called the conductance of a set of vertices or a set of proteins in the protein-protein interaction network. The conductance is defined as the ratio between the number of the edges from the set to the outside, the number of edges with exactly one endpoint in the set, divided by the number of interactions between the proteins in the set. And so one clue to find protein modules is to look for connected subgraphs with low conductance, a relatively small number of connections outside the set compared to the number of connections within the set. Now, another clue that we can use to find protein modules is that if they're important for the functioning of cells, they will tend to be conserved in evolution. And that means that a protein module in one species should be reflected by similar protein modules in other species. And so we have this notion of conservation of modules, finding the same set of, essentially the same proteins in some other species as you have in the module in a given species. So here we use the notion of homology between proteins. So we say that two proteins, either in the same or in different species, are homologous if there's evidence that the proteins have evolved from a common ancestral protein. And that evidence is most commonly reflected in sequence similarity, although one could look at other things such as similarity of functional annotation. And so we, instead of working just within a given species, say human, we might compare a human with Drosophila and look for conserved protein modules. In other words, a pair of modules, one in the first species and one in the second species, with the property that every protein in one of the modules has a homologue in the other one. And so we now formulate the search for protein modules given the protein-protein interaction networks of two species as the problem of finding two connected subgraphs of low conductance in the two respective species with homologous proteins, so that every protein in one has a homologue in the other. So now we have to devise a mathematical approach to finding modules first in one species and then transporting the information over to a second species. And that turns out to involve random walks on graphs. So the problem that we consider is to locally, somewhere in the graph, starting from a given initial vertex, to find a small set of proteins, a small connected set of proteins containing and fairly close to that seed vertex, which has low conductance. And this problem has been studied by a number of mathematicians and graph theorists, and an approach has been developed that involves random walks in graphs. So we're going to talk about two kinds of random walks. The first one is a kind of uniform random walk on a graph where at every step, the state of the walk is some vertex of the graph, and a random neighbor of the vertex is chosen, and the move goes to that neighbor. So it's just the most natural random walk on a graph. And it's known that in that random walk, the stationary probability of a vertex is inversely proportional to its degree. Now, we want to contrast that with a second kind of random walk, which is designed to explore the locality of some seed vertex. And this random walk is called the personalized PageRank random walk because it has application also to page ranking on the internet. But here we're using it for a different purpose. In this random walk, it behaves much like the uniform random walk, but with one difference, that there's a parameter p, which is the probability that at any given step it'll bounce back to the seed vertex from which it started. And that's called the personalized PageRank random walk, and it tends to be then more localized in the vicinity of the seed vertex. And theoretical work by a number of people has shown something along the following lines. If we define the locality of a vertex as the ratio between the stationary probability of that vertex under the personalized PageRank random walk, which remains in the locality of the seed vertex, divided by the stationary probability under our uniform random walk, which is just the degree of the vertex, then we can rank the vertices according to locality in order of this ratio. The higher the ratio of the local stationary probability to the global one, the more local the vertex can be considered. And there are theorems by Chung and Anderson and Dan Spielman and others showing things like this. I won't cite a precise theorem, but the tone of the theorems is that if the personalized random walk has vertices of high locality, then there will exist a set of low conductance obtained by taking, for some k, the k vertices of highest locality, highest ratio of local to global stationary probability, and considering those as the local set. And so by searching through this list of vertices of high locality and computing conductances of successively larger sets of vertices, one can find sets of low conductance in this manner. So now I can, using this as a tool, I can describe an algorithm developed by my student Lukman Hodgkinson and myself, in which we look for conserved protein modules in the following way. You start, and we do this many, many times from different seed vertices. So we're going to be, a typical protein-protein interaction network may have 10,000 nodes. So we're going to be starting these random walks from thousands of different nodes. And in each case, we use the random walk technique to find a set of low conductance near a given seed vertex, and then carry that set over by looking at the set of all proteins in the second species, which are homologous to proteins in the chosen low conductance set. And then we take the connected components of the graph induced by those homologs of the proteins in our set S, and we look at the individual components of that subgraph in the second species, and we test each of those components to see if it has low conductance and is comparable in size to the original set, and if so, then we will have found a conserved pair of protein modules. Now, this technique is weak in a certain sense because when we map over from the low conductance set S to the second species, we may get huge components. And we would like to prune those components down to sets of smaller size. And in the Produles program, we use some fairly simple-minded hacks to do that. So what I want to describe next is a more refined way of mapping over from one species to another. Any questions about the general approach so far? So one problem that I mentioned at this point is that we would like to find modules that are conserved across several species. And extending the methods that we use to that problem is an open question. So in order to refine this approach, I turned to some work that was done jointly with colleagues in Tel Aviv under the heading of topology-free querying of protein interaction networks. So here's the connection. The situation we're in is that we found a set of proteins in a connected set of proteins in one protein-protein interaction network, which we expect to be a module, and we'd like to find a corresponding module in the network of a second species. So what we would be looking for in the second species is a set of proteins, is a connected subnetwork in the second species, which contains at least one protein homologous to each protein in the original set S, and is of smallest size. So this can be translated into a pure graph theoretic problem as follows. You're given a graph and a set of colors. Now, what these colors represent is the type of the proteins. So here we're making the abstraction that homology is really an equivalence relation, that proteins fall into different classes, mutually homologous proteins, and these classes carry a common label that we'll call the color of the protein. So homology now is simplified to refer to two proteins in different species being of the same color. And so with this convention, or this simplification, we are led to the following graph theoretic problem. You're given a graph, a set of colors, and for each color, a set of vertices of that color. So in other words, every vertex in the graph carries at most one color. And now we want to find a connected subgraph with a minimum number of vertices containing at least one vertex of each color. So the interpretation is that the original, that the graph is the protein-protein interaction of a species. We're given the set of proteins that we would like to find within a subgraph, and we want a subgraph of minimum size that contains a protein of each type. And this is exactly the problem that Produles encounters in mapping over from a proposed module in a first species to a counterpart in a second species. Okay, so you've got colors, you've got a graph, and you want to find a smallest graph that contains at least one vertex of each color. And for simplicity, we'll convert this into a decision problem. We'll ask the following question. Given a graph and a set of colors and a labeling of each vertex in the graph with a distinct color, find a set of connected subgraph containing exactly one vertex of each color, or determine that no such subgraph exists. This problem can be extended to allow additional vertices so that you don't get exactly a perfect one-to-one matching. But here we'll ask for a perfect match. And this problem can be shown to be NP-hard even in the case of trees of maximum degree 3. So in the joint paper with the Tel Aviv group, a couple of algorithms for solving this problem were given. The first one is a dynamic programming algorithm. Is there any question about what the problem is? It's important to know what the problem is, so let me state it yet again. Given a graph, a color on each vertex, you're looking for a connected subgraph which contains exactly one vertex of each color. Okay, so dynamic programming operates in the following way. It builds a table associated with a certain predicate. The predicate takes as its arguments a vertex V and a set of vertices and a set of colors S. And the predicate is true if the graph contains a tree rooted at the vertex V and containing exactly one vertex with each color in S and no other vertices. And then it turns out you can write a simple recurrence relation which tabulates this function B of Vs for all pairs consisting of a vertex and a set of colors, building up in the usual fashion of dynamic programming from sets of small cardinality to sets of larger cardinality. And then it turns out that the predicate can be tabulated recursively in increasing order of the cardinality of S in time proportional to 3 to the power k, where k is the number of colors, times n, the number of vertices. So this is a convenient algorithm to use when the number of colors is small, but not very useful when the number of colors is large, which is the case of greater interest to us.
Is this only apply for the tree of degree 3?
No, for any...
The k is nothing to do with it.
No, k is the number of colors.
No, the 3. Number 3.
I think the question is where the...
No, the 3 just comes out of the recursion. It has nothing to do with the degrees of the vertices. A second very clever approach is due to Falk Hüfner, who was a postdoc in the Tel Aviv group with Ron and Roded Sharon and others, which formulates the problem as an integer programming problem. So what are we looking for? We're looking for a set of vertices X, which contains exactly one vertex of each color, and where X induces a connected graph. Now, the clever idea behind the integer programming formulation is the following, that if you have a connected graph, then you can designate one of its vertices as a sink, and each of the other vertices as a source with supply 1. And because of the connectedness, there will be a flow with a net flow of 1 out of each of the sources and a net flow of number of vertices minus 1 into the sink, which is and the flow is conserved at every intermediate node. Okay, so it then turns out, so the existence of this flow is a neat way of characterizing the property of the graph being connected. And so what we're looking for then is a set of vertices containing exactly one of each color, and in which, moreover, such a flow exists. And these conditions can be expressed by linear integer constraints with a 0, 1 variable to indicate membership of a vertex in the set X, and integer variables representing the flows in the edges. So this is a neat integer programming formulation which is useful up to a certain point. Now, in the application that I described, where we're constructing low conductance sets all over a huge protein-protein interaction network, and then trying to reach over to a second species to get the corresponding colorful subgraph corresponding to any given low conductance subgraph, we're going to be solving thousands of these instances of the colorful connected subgraph problem. And so we need a fast heuristic. And I've been somewhat obsessed with the heuristic I'm about to describe, and I've solved many hundreds of instances by hand, and it works quite well. But as with many heuristics, there's no formal proof of its general good behavior. So the idea of this heuristic is to gradually prune vertices away from a big graph until we can get a smaller connected graph which still contains at least one vertex of each color, and keep pruning away as long as possible. And the way we prune is to look at the frequencies of occurrence of the different colors and identify a fairly small set, let's say the 5 vertices of with the most frequent colors, the 5 colors that occur most frequently in the graph. And we'll focus on removing vertices of those frequent colors, as well as possibly removing vertices of the other colors to get a smaller graph which maintains the property of being connected and containing at least one vertex of each color. And we're just going to iterate this process. So what we do is, sorry for the typo here, what we do is we form the subgraph induced by the infrequent colors. And in that subgraph, we select a minimal set of connected components to cover all the infrequent colors. And then we reconnect the selected components using as few additional vertices as possible while including at least one vertex of each frequent color. So I need to give you a little bit of insight into how these steps are actually achieved. And the overall heuristic is to just repeat this process over and over again, pruning vertices away from the graph. And so, oh my. I see, something went wrong here, but let me try to explain. So if you ignore the letters in red, what you see here is an 8x8 array of letters. And these letters represent the different colors. I just generated this example by writing down a random piece of text. But we're going to view this 8x8 array as a graph. So this is just a convenient way of writing down a fairly large graph. The way we view it as a graph is just to assume that each occurrence of a letter is a vertex. The color of the vertex is the corresponding letter, and each vertex is adjacent to its north, south, east, and west neighbors. So what we have here is a grid graph. And by counting letter frequencies, we find that the 5 most frequent letters are E, T, A, O, and N. And so what we're going to do in the first step of the heuristic is to delete the occurrences of these frequent letters. And that gives us this subgraph. Now what we want to do is we see now that what we have left is the infrequent letters, and they break up into connected components. And we don't need all of those connected components in order to cover all the letters. So, for example, the letter S occurs many times, and so we can delete a component consisting of the single node labeled S in one particular place here. So the question is, how shall we delete connected components while continuing to cover all of the colors occurring, all the letters occurring? And here we exploit the fact that there are, in practice, very good fast algorithms for the Steiner tree problem. So what we will do is construct a Steiner tree on this set of connected components. So each connected component here is treated as a unit, as a single vertex. We construct a Steiner tree on this set of components, and then we look for components that we can remove while continuing to cover all of the different colors. We rank them according to how much the Steiner tree shrinks in terms of its number of vertices when the component is removed. So we continue to remove components, redundant components, as long as possible in a manner that tends to reduce the size of the Steiner tree. And when that can't be done any longer, we attempt to reconnect the remaining components with in this case, copies of vertices with the frequent colors. Oops, what have I done? Did I do that? Okay. All right, so this is what happens after these components have been, some components have been deleted and the remaining ones have been reconnected. The letters in red are the occurrences of frequent colors that are added to cover every one of the frequent colors at least once, and to, with a minimum number of color occurrences, reconnect the remaining components. So what we did was to prune away, in a manner dictated by the structure of a Steiner tree, prune away some of the unnecessary components of infrequent letters, and then reconnect using the frequent letters. And in this particular case, we're able to do it with only one occurrence of each frequent letter. And now we just keep repeating that process. So in this reduced structure, the frequent colors are S, R, D, and I, so we delete those colors. Look at the components that remain and try to reconnect them with additional vertices of the original graph. So this is what happens when we prune away the frequent letters. We see in this particular case, I think it turns out that there are no redundancies at all, that every connected component is needed. If we redo, as it happens in this example, accidentally I happen to construct an example where every one of these components is required. And then we reconnect as economically as possible with the infrequent colors. And we get this, where the frequent letters are N, A, and D. Now we can see by inspection that we can get rid of the N and the A at the top without disturbing connectedness. But we go through the process anyway of deleting all the occurrences of N, A, and D, looking at the remaining components and reconnecting them. And in this particular case, these are the components, and after reconnecting we get this structure in which the only repeated letter is D, and in this particular example further iterations will not improve, so this is the final solution. We didn't succeed in getting a perfect subgraph with exactly one occurrence of each color, but we had only one repeated color. So this, I have no theorems to offer about this, but this turns out to be a very effective heuristic, at least insofar as I can tell from many hours of amusing myself in front of the TV set doing hand computations. So another open problem would be to analyze the performance of the heuristic algorithm on a suitable set of random instances. Random trees might be an interesting example, or random grid graphs. So the work that I've described so far is captured in two principal papers, one by Hodgkinson and myself on the Produle's algorithm, and the other one on the colorful connected subgraph problem and its applications by several authors from the Tel Aviv group and myself. Now, in the remaining time, I want to talk about a second piece of work involving inferring the structure of signaling networks. So what is a signaling network? It's a directed graph. Each edge represents some kind of biomolecule. For convenience, I'll say proteins. So each edge represents a protein that participates in the signaling network or pathway. And we're going to take a Boolean model so that each protein dynamically, depending on the states of other proteins, is either activated or not activated. So each protein has activation state 0 or 1. And we'll assume that the logic of this model of signaling is given by a set of Boolean functions where there is a Boolean variable for each edge. And the activation state of any edge coming out of a vertex V is a Boolean function of the activation states of the edges pointing into the vertex V. And we will assume further in what follows that the directed graph is acyclic. So we're not modeling feedback here, although I think the approach can be extended to look at the extremely common case of signaling networks with feedback loops. So the directed graph will be taken to be acyclic. Now, the goal that we've chosen, and this is joint work with Roded Sharan, is to, given the graph, which is the pattern, essentially you can think of it as the pattern of causality, the pattern of interaction among the proteins. Given the graph, we want to use perturbation experiments to determine the Boolean functions. So we're assuming here that we have everything about the Boolean network except the logic functions that dictate for every edge how its activation state depends on the activation states of the incoming edges. And the perturbation experiments simply fix the states of a subset of edges and observe the state of, let's say, one additional edge. And that observation is called the outcome of the experiment. So this is a standard technique for trying to understand the logical structure of a network, where you clamp the values of some of the signals and see the effect of certain outputs. And in this particular case, we assume that each experiment looks at one output. Okay, so you're given the graph, you're not given the Boolean functions, but you are given the results of these experiments, each of which interferes with and fixes the states of certain signals in the network and observes the effect on one particular edge. And because the graph is acyclic, any choice of Boolean functions will determine what the outcome should be. And the optimization problem is to select the Boolean functions to maximize the number of outcomes that are correctly predicted, the number of experiments that are validated by the choice of Boolean functions. And a secondary objective, because it turns out that often there are many optimal solutions, is to minimize the number of non-constant Boolean functions. So if you can get a solution where most of the Boolean functions are not sensitive to their inputs, that's considered to be a preferred solution. So what we observed is something extremely simple-minded, but which doesn't seem to have been exploited before, namely that the problem of optimizing the Boolean functions to maximize agreement with the experiments can be expressed as an integer linear program. And the variables of the integer program correspond to entries in the truth tables of the Boolean functions. So if you have an edge which is influenced by 4 other edges, the truth table will have 16 rows, 2 to the 4th power, and you will need 16 0-1 variables, namely the entries in the truth table, to characterize the Boolean function. And then through some rather clever encoding techniques, one can write down all of the constraints imposed by these truth tables as integer linear constraints, where the number of constraints associated with each one of the edges of the network, each one of the Boolean functions, is 2 to the power k, where k is the number of inputs to the Boolean function. So if you're lucky enough that all of the Boolean functions have only 3 variables, then you would need only 8 Boolean variables per Boolean function, per edge. And so there are some cute tricks in the paper that mostly were constructed by Roded Sharan, showing how we can write down integer constraints that capture the effects of these truth table entries. Now, that's especially when some of the Boolean functions have high in-degree, this gets to be a pretty large integer program. So there's a special case in which we can greatly economize. The most common functions that will occur in networks are probably monotone. Ands, ors, inverters, majority functions. You may occasionally get something like an exclusive OR, but they're not too common in reality. And so we can focus on the special case in which if we take any one of these Boolean functions, any one of these edges of the network, and look at the influence of the other incident edges on its vertex, each of those inputs is known to be either an activator or a repressor, and the function value is 1 if and only if the number of activators in state 1 plus the number of repressors in state 0 is greater than or equal to a threshold. So it's just an ordinary threshold function, and that captures most of the common Boolean functions. Now, the advantage of restricting attention to this class of functions is that you can characterize the entire truth table of the function once you know which inputs are activators and which ones are repressors by a single threshold number t. And so one can write, in this case, an extremely compact integer program which involves as variables these thresholds. And so the problem comes down to inferring the thresholds from experiments. It turns out that the applications that we developed assume threshold functions. So we got fairly compact integer programs which in practice could be solved in almost instantly by a standard integer programming tool. And we were able to analyze a couple of interesting systems. So one of these is called the EGFR system, which regulates cellular growth, proliferation, differentiation, and motility. It has 112 molecular species, 112 edges, maximum fan-in 5. And the experiments measured the activity levels of 11 proteins in 34 conditions. And the integer program, assuming threshold functions, worked almost instantly, and it computed a solution that predicted 90% of the outcomes correctly, matching a curated solution that had been a hand-constructed solution. In a second example, the IL-1 system, which is involved in coordinating immune response upon bacterial infection and tissue injury, there are 121 edges, maximum fan-in 6, and again, the integer program correctly predicted the outcomes of about 90% of the experiments, matching a curated solution. So you might ask, well, how applicable is this technique going to be? It seems to work well, but the catch so far is that it assumes that you know the wiring diagram of the circuit. You don't know the Boolean functions, but you know everything else. And it takes a lot of work by biologists doing individual experiments to figure out all of these influences and get that wiring diagram. But what we've shown, at least, is that if they can get that far, then we can really come close to optimizing the choice of the Boolean function. So it seems to be a rather promising tool. And there are some open questions here. One is to give a method for the adaptive choice of the informative experiments. So we were assuming that somehow experiments had been chosen and conducted, but what you would really like to do is conduct the experiments sequentially using some criterion about the expected amount of information gained from each successive experiment. So we need some ideas about how to do that. We think that the integer programming approach can be extended to cyclic networks. Here you get the complication that there might be many equilibrium states of the network, even under fixed inputs. So there are some complications, but I think they can be handled. And we would also like to extend the integer programming approach to undirected networks where you don't know what the pattern of causality is. You don't know whether edge X influences edge Y or whether edge Y influences edge X. So all of those ought to be considered. And that brings me to the end of the planned discussion, and I'll be happy to take questions. Yeah.
I have one question regarding the background. When you mentioned the protein-protein interaction network, the current technology basically take one snapshot at a time, but the interaction is actually a dynamic process.
Yeah.
Actually, I'm wondering how confident you are or comfortable you are when you use the current data to say, wow, this is the subject I'm going to study.
Yeah.
One snapshot.
Yeah. So the question is, how meaningful are the protein interaction networks at present? And part of the problem is that the interactions are dynamically changing. We recognize that there is a very high level of false positives and false negatives in these networks. So in a way, we're looking toward the future where this can be improved. We have done some editing because it turns out that in some of these networks, the interactions that are included are predicted from other species rather than directly observed. And there are other complications, so that different experimental techniques can give incorrect readings. And so we realize that there is a lot of uncertainty about the information there, and we expect that this will improve. And we do find that we get some very good-looking networks, some of which conform to known modules, even with these handicaps. But things will be a lot better if the noise can be cleared up, and we're aware of that. Yeah. Was there another question? Yes.
Do you think you can infer the wiring in the interaction network from like gene-gene interaction network in some other way, without the experiments?
Without.
You mentioned that the biologists should do a lot of experiments to provide for wiring diagram.
Yeah.
Do you think we can infer these connections from genetic information, for example, from gene-gene interaction networks?
Yeah, I think so. So again, I think you need to. You probably cannot avoid doing perturbation experiments to know the direction of causality. If you know that two molecular species interact, it's hard to know which one is acting on the other. There are some techniques for doing that by sort of looking at signals introduced at one place and seeing how far they can reach into the network. But it takes a lot of work. So in a sense, we're solving only a piece of the problem, if I understand your question, because we're assuming that somebody else has done the hard work of figuring out the pattern of causality. Yeah.
Would it be meaningful to try to extend the situation to weighted networks, where you could distinguish between weak and strong activators, for instance?
You could look at multi-valued logics. The integer program could still be written down if we assumed, for example, that each protein had 3 states or k states. That could be handled with some complications. Yeah. Yes. Here. Yeah. Did you?
When you formulate a problem, finding a smallest group in the network,
yes,
which is homolog of the other group.
Yeah.
And my question is, why you have the smallest? And another way is, does it verify with the experiment, or the results? Do you have any experiment, The first question
Yeah.
is why it's small. The second one is there any verifications that the validity of your experiment, showing that it really works?
So let's, could you repeat the first part of your question?
Okay. You try to find a smallest set.
Yeah. Why smallest?
Why is the smallest? Okay.
It wouldn't have to be, but that seems to be the most. I don't know. It's a principle of Occam's razor that's the sparsest explanation that could be found. Yeah.
Okay, I know that this is we try to find optimal. You always. Another thing is when you find the results, do you verify that really? They do that.
Not in this particular study, but in past work with the Tel Aviv group and people at UC San Diego, we had some other methods which we used to transfer protein-protein interactions from one species to another. So in that work, we would look at conserved modules and predict that if an interaction exists in species A, it's likely to exist in species B. And we did get verification of those predictions with pretty good accuracy by talking a lab in Germany into actually doing those experiments. But we didn't go beyond verifying predictions of individual interactions. So we didn't have any way of verifying whole pathways, except to put them out there for the biologists to consider and get their opinions. Yeah. Yes, there.
Questions. The first one is just for knowledge. So in the beginning, you said that there were some results that connect locality to conductance.
Yes.
And it seems like the conductance is completely derived from the topology of the graph, but the locality has this parameter p, which would influence. And so I was wondering how you could make the connection without having the just connecting the topology versus something that depends on arbitrary defined.
Well, the parameter p has what we did in the experiments in the getting these statistical correlations was to generate several thousand conserved pairs across human and Drosophila. And how we did it is immaterial. We didn't, in fact, we did it in a simple-minded way that doesn't use conductance. But after the fact, we would look at these modules and we would compute a number of parameters: conductance, average degree, topological similarity of the two copies in the two species, number of different protein types. So various structural parameters, and we would try to correlate those against the strength of functional coherence using a measure derived from the Go hierarchy. And so then we would correlate modularity, which is this low conductance. It has nothing to do with how we found the conductance. We would correlate modularity, average degree, and all of these other things with functional coherence. And the two attributes that stood out as having a high correlation were modularity, which is low conductance, and average degree. And all of the other measures, including the topological ones, seem to have very little correlation with functional coherence. Yeah.
One more question.
Yes.
To the heuristic that you provide for the color.
Yes.
What's the you said that you could, it's not known even for trees. What is the simplest graph for which no sort of result?
Yeah, well, we know that even for trees, the problem is NP hard. We would need a model of random trees, and then we could sort of evaluate the results.
Do you have a conjecture for what form the result
No, not at this point. I don't have a conjecture about the form of the result. Yeah. Yes?
So, about this 3 to the k algorithm. So, if I remember correctly, there is also a 3 to the n algorithm for
Steiner.
coloring.
For what?
For graph coloring.
Yeah.
And it is also based on this dynamic property
Yeah.
of subsets.
Yeah.
So, but there are fast algorithms also, for example, based on inclusion exclusion.
Yeah.
So have you tried to apply this principle in your?
I think it's possible to get the exponent down, the base of the exponent down, but we don't really. I mentioned this in passing because it only works for small k in any case. But yeah, I think some improvement may be possible there. Yeah. All right then. Thank you very much.